I do, but I want to give everyone else a chance.
Rain, you like cracked yourself.
He completely cracked himself up.
Oh, I almost
So if you were paying attention to yesterday, camera's rolling, Tyler, what do you want?
I'm going to answer the question.
Seriously?
I was going to answer it.
Let's wait a few minutes. Let's let everybody kind of think about it. And let me drop a couple hints. Well, show of hands. The question is, which direction's water going to move? Is it going to move from the beaker into the bag or from the bag into the beaker? Don't answer out loud. Okay, so that's the question. You should know that based on what you see here. Okay. Remember, this is osmosis. Okay. That's the name of the process going on here. The movement of water from high concentration to low concentration. In other words, water's going to want to move to where there's more of it to where there's not so much of it. So you have to take those two different solutions. Hey, and by the way, the size of the solutions makes no difference. That beaker could have been the size of the room, and that little bag inside could have been the same size. That is not the factor here at all. The only thing you should be looking at is basically what those solutions are made of. Which one is more concentrated? Which one's more dilute? Then you can say to yourself, okay, the dilute one has more water in it. If the dilute one has more water in it, that water's going to want to move to where there's more of it, to where there's not enough of it. Show of hands. How many people know the answer? Confidently.
I think.
I know it 100%.
Okay. How many people have no idea what I'm talking about and want to admit it? Okay.
I know you know.
Tyler, since you're just, busting at the seams to give me an answer, and Grant, I got another one. You can answer that one.
All right.
All right. So tell me, is water going to move into the bag or out of the bag?
Go move into the bag.
No.
It's going to move out of the bag.
Wow. Very good.
I gave him a chance and he was wrong, so I answered it.
Put them
I wouldn't do that. I
away.
don't get that.
Okay? This is why. This is what you have to think about. Water inside the bag is a 10% sugar solution. Well, it's 10% sugar and like 90% water. A lot of water. The solution in the beaker is 23% sugar. That means it's only like 77% water. Less water. The stuff in the beaker is really concentrated. It's really, really sugary. The stuff in the bag, not so sugary. There's more water in the bag. Okay. Water is going to move out of the bag. Okay, definitely water's going to move out of the bag. Zane?
Put them away. It's in my pocket.
Food away. Thank you. Okay. Now my next question refers to a couple of vocab words we were doing yesterday. Is the solution in the beaker hypotonic or hypertonic to the solution in the bag? Grant, do you want to answer this one or do you want to answer the next one? That's a whole other question. You don't know this one?
No.
Remember the word hypo. What does the word hypo mean?
Under.
Under. The word hyper means above.
So it's going to be hyper.
It's hyper. It's hypertonic. It's a hypertonic solution compared to what's in the bag. Very good, because it has more solutes. Very good. All right. Same idea, slightly different form. Instead of percentages, I changed it. I have a 1 molar salt solution compared to a 3 molar salt solution. You really don't need to be concerned how to make them, but you got to understand that 3 is bigger than 1, and so a 3 molar solution will be more concentrated. It'll have more salt in it. It'll be a saltier solution in the cell than it will be in the beaker. So Grant, tell me. It's going to go into the cell. Very good. Water will move from the beaker into the cell. The cell will blow up like a balloon.
Hypotonic.
Okay? And that is a hypotonic solution. Very good. Good. See? Easy stuff, right?
Wait, which way will the water move?
Into the cell. Okay, let me draw an arrow.
Jeez, Leo.
No. This is important, and it's constantly flipped around in people's minds, so I want you guys to get it straight. It's going to go that way. Whereas the other one, water will move out. Remember, water's going to be the only thing doing the moving. the sugar is going to stay put. It can't pass through this membrane. That's why it's called a semi-permeable membrane. It can't pass through there. Only water is tiny enough to move through here, and only water is tiny enough to move through here. All right, moving along. I'm going to finish the chapter right now. It's not very many notes, and then once I'm done with the chapter, we'll do a couple of, it's more like little activities, to be honest. Yesterday's word of the day was passive transport. Today's word of the day is active transport. Active transport. Some things need to move by active transport. In other words, they're too big or they're not charged right to move in without energy. Energy is needed for active transport. And since I'm referring to the cell, you always want to think of energy in terms of ATP. So in other words, in order to make active transport happen, you better have some ATP around. Okay. Substances move from low to high concentration. Let me add something here too. Against the gradient is a fancier way of saying that same thing. In other words, remember in diffusion, like the perfume bottle, you were talking about perfume molecules going from an area of high concentration and spreading out? This is the total opposite. Here you have a situation where you have a cell. That circle is a cell. And you have some dots there that are representing, I don't know, some kind of ion. Who knows what it is? Like a salt, like a, let's just call it like a sodium ion. Okay, remember ions from last year, a sodium ion. Okay? Anyway, there are going to be times where you are going, imagine each of those dots is a sodium ion, Na+. You are going to want, this cell is going to want to push every last sodium ion out of it. In other words, even when it gets really concentrated with sodium ions outside the cell, don't care. I want to keep moving sodium ions out there from inside the cell. So every last one of those 3 sodium ions, bam, I want to push outside. That's what I mean by going to a low concentration inside the cell to a high concentration outside the cell. And that's another way of saying that is you're going against the gradient. The gradient will want to go have them slip back inside, but no. You're going against the gradient. So my next question is, what I'm doing is, I'm talking about a certain situation in organisms where you're going to see this happen. A very, very popular one to talk about when you talk about active transport is something called the sodium-potassium pump. Just have you kind of write this down. The sodium-potassium pump. Imagine this is one of your nerve cells in your body. Actually, this is what's going on inside your guys' nerve cells right now. All those neurons, all those nerve cells, making you think, conducting impulses to have you think, and to move your arms, and to breathe. Believe it or not, your nervous system does more than you think. A lot of nerve cells need to work properly there. If you take a nerve cell, in order to make it work right, you have got to have a really low concentration of sodium atoms inside the cell and a very high concentration of potassium ions in the cell. The little pluses here. I know this looks like it's technical, but this is actually a pretty easy concept. In other words, this nerve cell is going to want to pump as many Na sodium ions outside of it as it possibly can. Well, that's going to go against the gradient. You need some energy. That's active transport. Also, it's going to need to move through a transport protein. Hey, remember, parts of a plasma membrane. The only way these sodium and potassium ions are going to move is if they move through a special protein that is designed to work with ATP to push them out there. work for the cell to push all those sodium ions out there and all those potassiums in. Your nerve cells need to your nerve cells will be happy if this is what they look like before some kind of nerve impulse happens. In other words, a lot of people who have neurological disorders like nerve damage or nerves cannot have this happen. You know, maybe their sodium potassium pumps aren't working, pushing those sodiums one way or their potassiums the other. That can lead to some disorders. Okay, so that's kind of why this is necessary. Nerve impulses are, like I said, are needed for everything from breathing to talking to moving to everything. So, all right, and finally. I'm just going to write kind of abbreviated. I want to talk about bulk transport. Remember, this was that third form of moving things in and out of the cell. So I'm almost done. Bulk transport is a form of active transport. So basically, it's going to need energy. It's a form of active transport. You need energy. Moving large substances. All right, some things that a cell is going to want to take in or have it leave they're big. They're too big. They're too big to pass through those phospholipids. They're too big to even go through necessarily a protein. Okay, they need to be brought in a whole different way. Well, there's two different ways. I know these are big words. There's endocytosis and there's exocytosis. Here's your big words of the day. These are easy to remember, though. If something's brought in, if something is transported by bulk transport by endocytosis, it's coming into the cell. That's easy. Yeah, exocytosis, think about it.
No.
It's leaving.
Well.
It's going out of the cell.
Got it.
How easy is that to remember? Now let me give you some ideas of why you would need this. Let's imagine you are let's imagine you're a paramecium. A little one-celled organism floating around in pond scum, enjoying life. You're a little protist, one-celled critter. You gotta eat. All right? They're gonna spot a nice piece of algae. Literally, this is cool. I'm gonna kind of draw a picture of a paramecium. What this paramecium does is literally kind of like pinches in part of its cell, surrounding those little dots are food. They will literally kind of like reach in and grab and kind of like change its cell shape to ingest that food. It's pretty cool. It's pretty cool. It surrounds it and takes it in. That food's coming into the cell. They're probably going to take that food and head it over to a lysosome cleaner, where it's going to basically take that food and break it down so it can use the parts. You guys are filled with white blood cells. You guys know what those things do? They keep you healthy. How do they do that?
Like bars and stuff like that.
They hunt around looking for bacteria. When they find it, they engulf it. They bring it into them by endocytosis, take it over to a lysosome, and zap it. You're healthy again. Exocytosis. You're going to have situations where things need to leave the cell. Well, yeah, remember what cells do. Cells, their job in life is to make enzymes or hormones or other proteins. A lot of those need to leave the cell. A hormone will do no good if it stays inside of a cell. It needs to leave the cell and travel to where it is needed. Hormones are like the messengers in your body. They are made in one location and they move somewhere else. Well, if they move, they've got to leave the cell. I think that's it. I'll leave it at that. Questions on any of this? Pretty straight.